Stream ecology, also known as lotic ecology, is the study of flowing freshwater systems. Streams are important to study not only because of the ecosystems they support, but also because of their linkage to downstream rivers and marine ecosystems. This concept is best illustrated by considering streams at the watershed or basin scale. A watershed is an area of land where all of the incoming water, mostly through precipitation, eventually drains through the same location. For example, in Charlottesville, we live in the Rivanna watershed, which encompasses the city and parts of the surrounding counties. All streams in our area flow into the Rivanna River, which eventually flows into the James River. We can classify streams based on their origin. First-order streams originate at the highest elevations in the watershed. Second-order streams are formed by the confluence of two first-order streams. Third-order streams are formed by the confluence of two second-order streams, and so on. Note that a second-order and third-order stream do not form a fifth-order stream. The next higher order of streams are only formed by the confluence of two streams of the same order. The river continuum concept suggests that we can reasonably predict the abiotic and biotic conditions of a stream based on its order. It states that there is an orderly progression of predictably integrating dependent regions containing organisms whose ecological roles reflect the changes in river basin, geomorphology, current speed, gradient, sediment and organic matter composition, and allochthonous versus autochthonous production. The term allochthonous simply means energy and production comes from outside of the stream, and autochthonous means energy and production comes from inside the stream. This figure nicely illustrates the river continuum concept. The top of the figure begins as a first-order stream and presumably joins other tributaries to form higher orders as it widens. This figure seems to suggest that as stream order increases, the biotic community, which includes fish and macroinvertebrates, changes. Why might this be? In our stream ecology lab, we will be comparing the abiotic characteristics and biotic communities of lower versus higher order streams. In the field, we are going to measure discharge, temperature, and dissolved oxygen. Discharge describes the volumetric rate of water flow through a given cross-sectional area. Discharge is calculated with the equation above and is simply the product of water velocity and the channel width and depth. Temperature is directly related to the amount of incoming solar radiation and is strongly related to dissolved oxygen. Increasing water temperatures result in decreased diffusion of oxygen from the air into the water. Dissolved oxygen, or DO, is simply the concentration of molecular oxygen present in the water column and is measured in milligrams per liter. Oxygen in the water column is important because it is consumed by most organisms in the stream. Photosynthesis increases the amount of oxygen in the water, while respiration by other organisms, including fish, macroinvertebrates, and microbes, consumes oxygen, thus decreasing DO levels. We will also be sampling macroinvertebrate and fish populations to estimate diversity. In order to estimate diversity, we will use Simpson's D, which is determined by 1 minus the sum for all species and groups sampled of the number of sampled individuals belonging to one species or group divided by the total, then squared. So, for example, if you sample a stream and catch 9 crayfish and 1 dragonfly larvae, your Simpson's D will equal 0.18, which is quite low. This is an example of a lower order stream that might be similar to Steeker Creek. Note how narrow the stream is and the tree canopy cover and accumulation of leaf matter. How might these conditions affect temperature and dissolved oxygen? How will this affect photosynthesis rates in the stream, and what types of macroinvertebrates might you expect to find here? This is an example of a higher order stream that might be similar to the Rivanna River. Note how the stream is much wider and hence has lower tree cover and has much greater flow. How might this greater flow affect dissolved oxygen levels? What kind of microorganisms do you expect to find in these waters? For assignment 1, given what you have just learned about the river continuum concept, develop hypotheses for our study. Specifically, how do you expect discharge, temperature, dissolved oxygen, and macroinvertebrate diversity to differ between Steger Creek, the second-order stream, and the Rivanna, the fifth-order stream? How do you expect the season to affect our findings in the stream, i.e., how might sampling in the fall compare to sampling in the spring or summer? For the first assessment, submit your hypotheses, which should be at least 1 to 2 paragraphs on Colab or at the beginning of lab, depending on your TA's preference. Do not simply regurgitate this table. Explain why you would expect to find such differences and what kind of relationships might exist between these parameters. For assessment 2, download the Dino Diversity Curve example spreadsheet and then complete the exercise. Instructions are provided in the spreadsheet.